Welcome back to our chapter on microbial growth. Here's what you're supposed to learn in this chapter. Classify microbes into 5 groups based on the preferred temperature range. Identify how and why the pH of culture media is controlled, and explain the importance of osmotic pressure to microbial growth. So let's start with general growth requirements. Every microbe has its preferred temperature, pH, and osmotic pressure. Those are physical requirements for that particular microbe's growth, and it's quite different for different microbes. Chemical requirements include a carbon source, nitrogen, sulfur, phosphorus, in various amounts for various microbes. Some require also trace elements, oxygen, organic factors. Those would be chemical requirements. So on the physical requirements, each microbe has a minimum growth temperature, an optimum growth temperature, and a maximum growth temperature. So the minimum growth temperature would be the minimum temperature that's absolutely required for this microbe to be growing at least a little bit. Optimum is where it grows the best, and maximum is the highest temperature by which it can still grow, but growth is probably be limited at that temperature. So here, on the temperature requirements, there are some terms that you need to know. Cold-loving bacteria, so those that like to grow in your refrigerator, for example, those are called cyclophiles. They love it cold. Phile comes from the Greek and means to love. Mesophiles, they like middle temperature. Meso means middle or moderate temperature in this case. And thermophiles, those are the heat lovers. They like hot temperatures. So here are some examples, and you need to know all of these terms. So cyclophiles, those are the real cold lovers. They grow best between minus 10 degrees Celsius and the maximum growth temperature will be 20 degrees Celsius, which is about room temperature. The cyclotrophs are the ones that are most dangerous because they really like to grow at refrigerator temperatures. So it's kind of tricky. So cyclotrophs, they like it cold, but not as cold as the real cyclophiles. Cyclotrophs, they have a growth maximum around room temperature. They grow quite well in your refrigerator, which can be a problem. And mesophiles, those are the moderate temperature lovers. They start growing at around 10 degrees Celsius and then up to maybe 50 degrees Celsius with a temperature optimum right around body temperature, around 37 degrees Celsius. Then we have the hot lovers, the thermophiles. Those are start growing around 40 degrees Celsius, so that's a little bit above body temperature, and then they go all the way up to 70 degrees Celsius, which is water that is so hot that it will hurt if you put your hands in. And their optimal growth temperature is about a little bit above 60 degrees Celsius. And then the real hot lovers are the hyperthermophiles. They start growing around 65 degrees Celsius and go all the way up beyond boiling temperature. So these guys might survive an autoclave, which could be a problem, but luckily those hyperthermophiles, those are archaea and they don't make you sick. So here is a text slide about the temperature requirements. Cyclotrophs grow between 0 degrees Celsius, so that's freezing on the Celsius scale, and then 20 to 30 degrees Celsius, and those are the ones that can cause food spoilage. So go back to this one here, the cyclotrophs. These guys here are a problem. I'm sorry, here from here to here, these guys, something to watch out for because they can grow quite well in your refrigerator. And here is a slide that shows you food preservation temperatures. And so let's take a look at the Celsius scale here. Above 60 degrees Celsius, and this is the range that destroys most microbes. If you go right around 60, it will take quite some time. If you go autoclave temperature above here, then you can destroy them pretty quickly. Most bacteria grow quite well in that range between 15 to 50 degrees Celsius. So that's called a danger zone because bacteria grow very well in this temperature range, and some may produce toxins. And so if you keep food at that temperature, that can be a real problem. At refrigerator temperatures below 15 degrees Celsius, so right in this kind of lighter red range, many bacteria survive, some may grow, and that can be a problem if you're refrigerating stuff and then expecting that it doesn't spoil, and it may still spoil. Now, if you're putting stuff in the freezer, then you should not get significant growth. So that's a good way of preserving food items, as you know. Let's take a look at this here, the effect of the amount of food. Let's say you made a big pot of something. Here the example is given with rice. Now, if you have a large pot with a large volume of stuff in it, like this red pot here, then it will take quite some time. Even if you put this pot of rice in the refrigerator, you can see that it's entering, so you're putting it in the refrigerator after boiling the rice, and then you can see that it will take it will take some time to reach temperatures, actual refrigerator temperature. So you have here 5 hours before it actually, the center of the pot reaches the refrigeration temperature. Meanwhile, the bacteria could be multiplying in your rice, and that could be a problem. Now, if you have a smaller pot with a smaller volume, then it reaches the refrigeration temperature much faster. You can see that right here. So it's entering about here, and it's already in the refrigeration temperature right here. So it's only about an hour where it's in the danger zone versus 5 hours here. So something to keep in mind, if you need to refrigerate something, be aware that a large volume of food stays at the danger zone much longer than a small volume of food. Let's take a look at the thermophiles. Optimum growth temperature 50 to 60 degrees Celsius, found in hot springs and organic compost. Hyperthermophiles, most of which are archaea, they have optimum growth temperatures above 80 degrees Celsius. They can thrive in boiling water. You would find them in the hot springs in Yellowstone National Park, for example. So here, so this slide shows you a deep sea hydrothermal vent. You have giant tube worms that are visible, and water is being emitted through the ocean floor at temperatures above 100 degrees Celsius, which is above boiling temperature, and you can still see that there is this microbial biofilm that's forming with these hyperthermophiles that are thriving actually at those temperatures. Now let's take a look at the pH requirements next. Most bacteria, they love a pH right around neutrality. So right around 6.5 to 7.5, and 7 being the point of neutrality, of course. Molds and yeast, they like a little bit more acidic environment, so they like to grow between pH 5 and 6. And then we have acidophiles that really like an acidic environment, but that's rather rare. Most bacteria right around neutrality, and yeast, keep that in mind. Yeast and molds, those are fungi, they like it a little bit more acidic. As far as osmotic pressure is concerned, there are not many organisms that can thrive in a hypertonic environment. So let's take a look at what happens when you are placing an organism in a hypertonic environment. That means we have a high concentration of dissolved particles in the environment. And so that usually causes plasmolysis due to high osmotic pressure. Now, there are a couple of bacteria that can withstand that. Those are extreme halophiles or obligate halophiles. So these are archaea again. They actually require high osmotic pressure to thrive in that environment, but most organisms will not be able to grow in a high salt concentration. Therefore, high salt concentrations are quite a good preservative. There are some facultative halophiles that can tolerate high osmotic pressure, but it's rather rare. Let's take a look at what this looks like. So plasmolysis is the situation where a bacterium here is placed in a hypertonic environment. So we have here, let's take a look at this over here. We have high salt concentration, 10% in the environment. And remember, water goes down a concentration gradient. So if we have 10% salt here, that means we have 90% water, 90% H2O. Inside the cell, we have 0.9% salt. Therefore, we have 99.1% water. So 99, let me put it down here, 99.1% H2O inside the cell. So where's more water? Well, obviously 99.1% is more water than 90%. So water will go down its concentration gradient, and we call that osmosis, diffusion of water. And as water is leaving the cell, it creates this sort of dehydrated state of the cell interior. Many organisms, if they can form a capsule, if they can form a spore, they will do that at that point. Other cells, they will just die, or they won't be able to grow, at least, to the very least. A normal situation is where you have the cell in an isotonic environment where the water concentration inside the cell and outside the cell is the same, and the cell is all happy and will be able to grow. So knowing that what happens in high salt concentrations or also high sugar concentrations, you can use that knowledge to preserve food. So for example, pickled stuff that's in high salt concentrations, that's a natural preservative. Or if you have high sugar concentrations like in jams and jellies, then you can preserve your food quite well, because most bacteria cannot grow in this kind of environment, and therefore, as a result, you have a natural preservative. But if you figure that you're going to reduce your sugar concentration to save calories, and you're reducing it beyond below a certain level, you will find that your jams and jellies, they will actually no longer preventing the growth of bacteria, and then therefore it will spoil much faster than before. So now let's take a look at some chemical requirements. Carbon source, that is a requirement for all bacteria except those that are autotrophs, that can make their own carbohydrates by photosynthesis. So any organism that cannot photosynthesize, you need to have some organic molecules available for food or for energy. The chemoheterotrophs, they use organic molecules as energy, so that's most of the bacteria. And autotrophs, they use CO2 from the air, just like plants would, and they make their own food. Now, nitrogen requirements. Nitrogen is a component in the nitrogenous basis of DNA, any nucleotide-based molecules such as ATP or NAD, any of those nucleotide-based molecules, they require nitrogen. Most bacteria, they decompose protein material for their nitrogen source. Some bacteria, they can use ammonia or nitrate from organic materials, and a few, they can use gas nitrogen and then do something called nitrogen fixation, where they use the gaseous form of nitrogen and fix that into solid compounds. Sulfur is required for some amino acids. Then thiamine and biotin also require sulfur. Most bacteria decompose protein as their sulfur source, but some also can use sulfates or hydrogen sulfide, which is a pretty smelly gas. Phosphorus requirements. Cells need phosphorus to build DNA, RNA, ATP. Many other molecules require phosphorus, so you can find it in membranes. And phosphate is a source of phosphorus for organisms. Then we need some trace elements, usually depending on what exact organism it is, it can vary. But there are usually some inorganic metal ions that are required for enzymes to work. They sometimes are cofactors for enzymes, and so those include iron, copper, molybdenum, zinc, and others. Oxygen. Let's take a look at the oxygen requirements, and that is something that we will continue on the next video.